Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes, and this is a biology lectures for the 11th class students. Since past so many weeks, I'm not very consistent in uploading any of the videos, and since we had given a timeline that we will try to complete everything for your NEET 2026 by November on this YouTube channel, so that later on we can actually work on fine-tuning the things and enhancing the practice sessions with the all the different kind of questions, short notes, mind maps, and everything's after that. So from now on, I will be regular with the uploads of the video, so you can rely on it. Meanwhile, all these notes which we are making throughout this lecture, that would be available on your on my Telegram channel. The link will be in the description, as well as on the WhatsApp channel. The links would be provided for the notes. Let's just start with it. Today for the 11th class, we're going to be starting with another chapter, that is anatomy in flowering plants. And anatomy in flowering plants, why we are studying this chapter? The major reason for studying this chapter is that since the angiosperms are currently at this time when we are studying in this era, it is the most abundant type of most abundant type of plant. Okay, so you know that what are the different varieties of plants divisions which we have studied? We have studied algae, we have studied bryophytes, pteridophytes, gymnosperm, and angiosperms. Out of all of them, which is the most apt and most abundant presently at this time? That is what? That is your angiosperms. So that's why it's very important for us to learn about them, to see about them, and maybe to extract the benefits of them. Okay, so our chapter for today is anatomy of flowering plants. Now, in this chapter, we're gonna be dividing this chapter into two different parts. So in current part, we're gonna be studying about the basics, and in the tomorrows, we're gonna be seeing that how we will be applying those things. What is basics? Anatomy means what? Anatomy means how a body is made up of. We know that all the living organisms, are basic unit is what? Cell. So all are made up of what? Cell. So cells are what? The basic unit, about which we're gonna be soon studying in the next botany topic. Okay, then these cells, when the group of cells are present, group of cells, what kind of groups? Either they are, first of all, they should be structurally similar, and performing similar function. Means those cells are called as what? They are called as your tissues. Okay, when the group of cells, such cells are there, we call it what? Tissue. Now these tissues come together, and different tissues are coming together, and they forms what? Not the organs. They're gonna be forming what? Tissue system. So in case of plants, from tissue we goes to what? Tissue system, not the organ or the organ system. Directly we reach to what? Tissue system. Okay, like for example, in our body we have what? Digestive system, reproductive system, excretory system, circulatory system. Same, they also have the tissue systems present. Okay, so this is what we have to study. So we know what we have to study about the cells. Okay, we have to study about the tissues and the tissue system. In this chapter, we will be focusing, like in this video, we would be focusing on the tissue part of it. Tissue system part, we're going to be studying in the next class. That's how I will destroy my pencil. Okay, so next we're going to be studying in the second chapter. Second part of this chapter would be what? Tissue system. Now, if we are studying about it, so there are the terms like this: anatomy. What is the meaning of what? Anatomy? Anatomy meaning that it's the study of the internal structure of the plant. Okay, so anatomy means what? Anatomy is the study of internal organs or parts of the organism. And what is morphology? Outside part, that outside appearance of organisms, how it looks like. So that is the two differences. Moving on to the next thing, that is what? That is our tissues and how they are made up of. Within the tissues, they are made up of different types of cells, and the cells have different naming present. So with this approach, we're gonna be studying. So we're gonna be starting with what? We're gonna be starting with the topic tissue. Tissues. Till now we know what tissue is. What it is? A group of cells with a common origin, structurally similar, or functionally doing the same work. Okay, so tissue is what? It is a group of cells having common origin and usually perform a common function. Okay, let's just move a little more. That if we are saying that they have a common origin, means they are made up of similar kind of a cells, or the cells might have even modified a little. Okay, we have learned this thing, a modification of root, stems and leaf, because we know that where that part is originated from, that's why we are dividing into radicle, plumule and all those things. If you haven't seen it, go back and see that video. You will understand it much better. And what are the different types of tissues that could be present? So basically they are divided on the basis of whether they have a capacity of dividing or not. Before going to that, let's build a concept. Okay, so this topic now it's coming up, what? Concept builder. Concept builder. When we are studying about the concept builder, so there are terms we have to remember. Okay, but as we learn it, we need to know that how a cell behave in the plants or in even organisms. Okay, that would be the better word. So basically what happened, there is a cell, like initially when we are forming, our cells are continuously dividing so that it increase our cell number so that it can form various cells. Like for example, imagine that you're all of us, we have studied in 10th class that the life starts from zygote. The zygote turns into fetus or the embryo. So zygote is what? Single cell. The single cell has to make multiple cells. So means what is happening? The single zygote cell is dividing. All the cells that are forming, that are dividing again and again. So these cells which are dividing again and again to increase its number, okay, these cells are called as what? In case of plants, we call it meristematic cells. Okay, so we are saying what? We are telling the story of a meristematic cells. So these are what? They keeps on dividing. We call it what? Meristematic cell. Okay, one point we have understood. Now this meristematic cell, like if we keep on increasing, means it will what? It will make so much of cells. Certain cells has to do certain part. For example, in my body, when my body will be forming, certain cell has to make the brain, certain cell has to make my lungs, certain cell has to make my hands. So every cell has to be specialized in its work. So what happened? These cells, few of them, okay, few of them keep on dividing, but few of them, they do what? They stops dividing. Okay, they do what? They stops dividing and acquires a permanent function. Function of what? Maybe making my brain or all those things. I'm giving you an example like this. Now that cell is called as permanent cell. Okay, so I will say that this cell, they form a group of cell over here, and these are what? These are my permanent cells. Okay, first statement we understood. Now later on what happened, these permanent cells says that okay, I will I don't want to keep on being a permanent. I want to again divide. So certain of these cells, let's say that this cell, it starts again dividing, starts again dividing. And you know that I told you what initially I told you that if any cell which is dividing, we call it what? We call it meristematic cell. Okay, what kind of meristematic cells? That's secondary thing. Okay, so it will what acts like meristematic cell, because meristematic cell is only what I have told you that it will divide. Okay, so this is one cell that is forming, that it started again dividing. Okay, later on what can happen? It can stop dividing again. So this process keep on happening in the organism body. Let's just remember the facts now. What are the facts we have to remember? That this process from a meristematic cells to this means it acquiring a permanent function. We call it what? This process is called as differentiation. That the cells earlier which were just dividing, which do not have any function, it has acquired a particular function. This phenomena is called as differentiation, that it has differentiated its cells from the meristematic cells. First point. Now what happened? This permanent cell, it did what? It stops its permanent functions and again formed what? Again formed meristematic activity. Again, this process means what? It lost the capacity of dividing, means it lost its permanent, sorry, not the lost the property of dividing, it's lost the property of its permanent function, lost its permanent function. And this is called as what? This is called as de-differentiation. D means deleted. Okay, I call it what? D means deleted. Delete the differentiation. If you will delete this step, remember, delete this step means what? It again goes back to this kind of feature. So this is de-differentiation. But I also told you that sometimes what happened? This cell, it again lost the capacity of dividing and acquired a permanent function second time. Okay, again lost the capacity of dividing, acquires permanent function. So what happened? It is differentiated, but here we call it what? Re-differentiated, because already this cell was, during its life course, it was differentiated one time. If it is again getting differentiated, we will call it what? We will call it this phenomena as re-differentiation. Re-differentiation. And now it has what? It has permanent function again. So it is a permanent cell. Okay, so what I told you three things. There is differentiation, de-differentiation, and re-differentiation chronologically. Okay, what is happening? Not every cell will do it, of course. Certain cells would be what? They will stop at this point. But certain cells goes through this whole way. Okay, so that could be there. Now one more thing. Now over here what I'm saying this is a meristematic cell. This is also meristematic cell. This is permanent cell. This is also permanent cell. Confusion? Okay. So we will do what? We will make it little easier. How are we going to be making it easier? First of all, the first meristematic cells which is initially being formed, we call it what? Primary meristematic cell. First time that is forming. Okay. And later on, again when it is forming meristematic cell, this we call it what? Secondary meristematic cell or tissue. Okay, then we have what? This permanent tissue. This is what? This is the primary permanent tissue when this is forming directly by differentiation. But if it is forming by re-differentiation, we call it secondary permanent tissue. Understood? So what is the concept? The concept is simple. Here I am highlighting it. So what it is there? We have primary meristematic tissue. What will happen with the primary meristematic tissue? It will undergo what? It will undergo differentiation. If it will undergo differentiation, it will form what? It will form primary permanent tissue. Later on, what will happen? This primary permanent tissue will undergo what? It will again undergo what? The capacity, again regains the capacity of division. We call it what? It lost the capacity of differentiation, so de-differentiation. So we have deleted the differentiation steps and again reached to the initial point. And here we call it what? Here we call it secondary meristematic tissue. Okay, moving on to the next. Sometimes this secondary meristematic tissue, it will again what? Loses the capacity of division and again form a permanent tissue. This would be what? Secondary permanent tissue. And this process is called as what? Redifferentiation. Okay, this will again be coming this topic in your plant growth and development topic, but here it will be what we need it so that we can understand that what is the growth pattern looks like when these cells will be seen. Okay, so this is there examples, we will learn it. For now, just remember that there are what? These are different types of cells and they are forming during the due course of time. Okay, moving on to the next thing. Now we're going to be studying about the tissues. So from now we have understood that there are two types of tissues, meristematic tissue and permanent tissues. Okay, so we're gonna be classifying the tissues broadly. Okay, so we have what? Classification of tissue. Classification of tissues. Now when we are classifying the tissue, we know that there are what? Two types of tissues on the way of their function, whether they are capable of divide or not. So we have what? We have meristematic tissue or we have what? Permanent tissue. Permanent tissue. Now the meristematic tissue is what? It is capable of division, can divide. Okay, permanent tissue do not divide, have permanent function, like certain cells which are having chloroplast, they will do what? Photosynthesis. Certain cells which have a large vacuole, they will save certain materials inside it. Okay, certain will have secretory functions, so these things will be there. Now this meristematic tissue, till now what we have said, we have divided them on the basis of what? On the basis of that what is their timeline of the development. We divided into what? Primary or secondary. Okay, so we have divided it what? On the basis of what? How they are being forming. Okay, so there could be primary meristematic tissue or there could be secondary meristematic tissue. Okay, then primary meristematic tissue do what? It forms primary plant body. So initially that would be present, that will increase the size of the plant. It would be what? Primary meristematic tissues. So forms primary plant body, primary plant body, and it is what? It is responsible for the vertical growth of the plant. So initially you might have seen that certain plants are there which do what? They grow like this, but they are very thin like me. But later on, they over the period of time, they will becomes what? Larger, thicker in the diameter. Okay, so they do what? We can say that responsible for the vertical growth. Okay, secondary meristem is what? Secondary meristem helps in forming the secondary growth, and it is not present in all. Okay, it's present in dicots and it is seen in case of gymnosperms. Okay, so it is what? This is forms secondary growth, forms secondary growth, often called as horizontal growth, seen in here. This is seen in all plants, seen in dicot and gymnosperms. This you have to remember. Now, since we are talking about the tissue, the tissues would be certain names to it. Now, if you have remembered that primary meristem, at different positions they are present and their different names are given. Okay, so we will remember that what is the primary names that is being given to it. So for example, the name that is given as like shoot apical meristem, root RAM is root apical meristem, and root apical meristem. Okay, we have axillary buds. Okay, so we have what? Axillary buds. Okay, along with that we have cambium. These are present. Okay, now secondary meristematic tissue. Here examples are what? Here the example, it is also called as lateral meristem. Okay, lateral means laterally present, later on comes. So it is what? It is the examples are, certain examples are, not all the examples, certain examples are like we have interfascicular cambium, we have fascicular cambium, we have cork cambium. These three you have to remember. So what are they? Cork cambium, fascicular cambium, and interfascicular cambiums. That's it. Now same we have studied about it, same goes with the permanent tissues. Permanent tissues are what? They are divided on the basis of primary permanent tissue, or there is secondary permanent tissue. The primary permanent tissues. The primary permanent tissue is the one which is directly formed by the differentiation of the primary meristem. We remember this flow chart. Okay, so we can simply write it what? That forms by differentiation. Everywhere it is common. Differentiation of primary meristem. Okay, and secondary will be formed by what? Formed by redifferentiation of secondary meristem. Okay, so primary permanent tissues like your what? Your xylem and phloem that is being formed. Okay, primary xylem and phloem, secondary xylem and phloem, or maybe the cork cambium is forming the structures like your bark and all those things. That is that whenever they are being formed, that is a part of the secondary, which we will learn it since there is no space. So we are not explaining it over here. We will explain over the period of time. Okay, of course, these things will again come up in the plant growth and development topic as well when we're going to be studying about these things in more detailed form. Okay, now let's just start directly to the meristematic tissue first, and then we will go to the permanent tissue, which is our chapter. So our chapter is divided into meristematic tissue and the permanent tissues. So first we're going to be studying about what? First we're going to be studying about the meristematic tissue. Meristematic tissue. Now in meristematic tissue, we know that the growth of the plant is largely due to this tissue only. Okay, because it will increase the number of it. So now we're going to be starting with the meristematic tissue. Already we know that there is a primary or secondary type of meristematic tissue. Now we're going to be studying that these meristematic tissue is doing what. Finally we know that it's making our initial plant body. As it is making our initial plant body, we need to know that how the cells will look like. Now these cells are rapidly dividing. So what happened? Their nuclear content is rapidly dividing. Means dense nuclear content would be there. Moreover, their cell wall would be thin enough. It won't be thick enough because thickening of the cell wall means will be a mature feature. Then vacuoles would be not present or very small, where normally they are not present into it. So vacuoles won't be seen over there. Okay, they will not be doing any permanent function. Okay, they will be just function would be what, dividing, and the rapid growth of divisions would be seen in their cases. Okay, so these things would be there. So there won't be any resting phase in their life. So if we see that cells will be what, they will be having dense nucleoplasm, means their nuclear thing would be densely present, okay, as majority of their time frame. Okay, then cell wall will be thin. Okay, next goes to there will be no less vacuoles present. Basic features of these cells. Then the meristematic cell, okay, it basically means what, divided. Okay, so it means that they are dividing cells. So it names itself says that dividing cells. Dividing cells. Okay, M for meristematic, M for multiplying. Multiplying means keep on increasing its number, right? So remember these things. Now at the different locations they are present and their different name and the functions would be there. So that is the second way of dividing a meristematic tissue. First way was what? First was that whether it is at what stage of development it is with respect to differentiation, de-differentiations, right? So here it would be what? It would be on the basis of the location where it is present. But which meristem? We are normally talking about the primary meristems. Okay, so primary meristematic tissue is classified as follows on the basis of location. So there are three different locations that we will be talking about. Okay, number one is what? Apical meristem. Apical meristematic tissue. So apical means what? That they are present on the apex of the cell. So what, so of the plant body. So there is a root apic meristem or the shoot apex meristem. So it means what? Located at apex of the plant body. So there are two different opposite part of the plant body. One is the root apex, other is the shoot apex. So there will be root apical meristem and shoot apical meristem. So there are what? There will be of two types. We have what? Shoot apical meristem and root apical meristem. Now here you have to remember that NCERT says that they are present at the tip of the root or tip of the shoots or the distant mode growth part of the shoot. But in reality, the root apical meristem that is present at the apex of the shoot. But the root apical meristem is present little subterminally. Okay, apex or the terminal. This is not present terminal because if you remember the morphology of flowering plants where we talked about the root zones of the roots, regions of the roots, in that when we were studying about zone of meristematic activity, Okay, so there we have said that root cap is there and above to it is present. Root cap is what? Providing protection to it. So means that the activity, this meristematic activity is not present at the tip. Like for example, if this is your plant, so the shoot apical meristem is present here, but the root apical meristem would be somewhere over here. So what does that mean? That means that it is present subterminally, little. Okay, NCRT says that it is present terminally, but correct way would be what? Correct way would be subterminally. Okay, so they are present at the subterminally as protected by root cap. Okay, so this thing is there, and that's it. This is about the apical meristems. Then what happened when the shoot is growing? At certain point, few of the cells left behind, and then it keeps on growing, which means what? That certain things left behind, those certain things which left behind, they are called as intercalary meristems. Okay, so what is happening? Shoot is growing. While growing, certain meristematic cells are left behind, trapped behind, something happened, and most of them will keep on growing. So these left out portions or left out group of tissue is called as intercalary meristem. And how important is this intercalary meristem? This intercalary meristematic tissue will help in the lateral growths. When I'm calling the word lateral growths, not that the lateral date, lateral in terms of the time I'm saying, means for example, it occurs in cases of grasses. So it happens in grasses. So what happened grasses? The grasses are being eaten up by the herbivores. So for example, I am the one herbivore and this is the one grass. Okay, so I go and eat it. Later on, I will leave it and I will go and eat the other grass. Okay, so what happened? I am not eating whole grass. Now the first question, why as a herbivore I am not eating the whole grass? Because these grasses has certain tissues which will do what? Which will bleed or make a cut on their gums. So they will like at the herbivore I go, I eat it and I got a cut. I was like, okay, this grass is not good, so I will go other. I will do the same on the other grass. Other grass is also doing, I am going. So I am a dump. Don't get me don't get offended by that our herbivores are dumb, because later on you will cut this and put it somewhere and all the people will come and being intolerant come and gonna bash me on this YouTube video. But no, that's not the case. But I'm just saying that to remember that the grasses has this what intercalary meristem, which do what? It basically makes our plant to regrow. Because what happened, if it will eat up, so only little bit of grass is left. Now the grass will have to grow again in his life. Okay, just think that if you will cut this part of it, I want to grow this part also. Of course it can't grow up, so I want to grow it because it will help me to sustain a life, right? So grass will also do the same thing, so it will grow again, and that is known as what? Intercalary meristem. Okay, so in grasses grazed by herbivorous animals are regrown due to these meristematic tissue. Understood? Now the last one is the axillary buds or axillary meristem. Now what happened? Certain, like when there's like the stem, like for example the root is growing, certain cells are left behind to the axis of the body, and from there the branch, flower or leaf arises. That is called as axillary bud. Since it is present close to the axis of the body, we call it axillary bud. Okay, so during the formation of leaves or the elongation of the stem, some of them left behind. Okay, so this happens is during the formation of leaves or elongation of stem. certain cells left behind forms later branch petiole of leaf or other thing or the flower. Okay, so this is what three things which you will be having. Remember, now with respect to this, one diagram is given in NCRT, so we're gonna be making that, which is diagram representing the shoot apical meristem. Okay, root apical meristem we are not doing now, we will just do it at the end. Okay, so the shoot apical meristem diagram was something given like this. So we have this part of the shoot. Okay, and over here we are they are growing their leafy part of it. Okay, so there is this is a growth curve of a leaf primordia. Okay, primitive leaf part. Okay, so this is what leaf primordium. Okay, future leaf will be formed. Primordium, one primitive pre-formed structure. So initially the leaf will be forming like this roll kind of a thing, if you have seen closely the new leaf. Okay, so this is leaf primordium. In this leaf primordia, where this part, lowermost part is present, here you will see what? You will see a meristematic tissue. And this tissue is considered to be what? It is considered to be the shoot apical meristem. Okay, so here what happened? This is the shoot apical meristem. Because if you have seen any plant, so plant growing like this and the leaf is also, it is not like that leaf coming laterally. Imagine that this is the topmost part. So in this topmost part, these are the leaves, my hands are the leaf, and this part which you're seeing over here, that is the apical meristem, beta. Now when the leaf will unroll like this, right, and this part will grow further. Understood? Imagination is done. Okay, so this is what I have made it. NCRT also made it. Now certain parts of it might left behind, like for example over here. Okay, so imagine that this is the certain part of it which is also present. Okay, which will be grow. So this part, if you see over here, this is what? This is the axillary bud, which is present close to what? Body of the plant. If you see that this is the main axis of the plant body. So from here it is present. So this is what? Axillary buds. Okay, and later on from this structure there will be other tissues that will be present over there. Okay, so this is what? This is showing your shoot apical meristem. Root apical meristem, I have given you example from the chapter number 5. You can go back and see that video and you can find it very easily. With this, we have completed all part of the meristematic tissue. Okay, so in your NCRT, meristematic is divided into two major parts. That is what? Primary meristematic tissue and the secondary meristematic tissue. After that, we have studied what? We have studied that there will be what? Permanent tissues. Look, this topic we are just doing in this video from the NCRT perspective, not going beyond to it. Because when the plant is developing, there is one more topic that is being is taught in the coaching institutes. What are those topics? That topic is that what are the theories for the histology. Histology means how these cells are forming. So that theory is given to you. Okay, so there are Derman theory and so many theories are there. We will be not doing it. A different perspective video will be given to you if you want to take a special information later on from this topic. For now, let's just keeping and sticking ourselves with the NCRT, because already these things are being deleted from the new NCRT. So these are the topics which are still there in your syllabus, coming up in your paper examinations, given in your MNC module where the syllabus is written. So we are going to be sticking with the NCRT only for that, not going beyond to it as of now from this video. Okay, so first part of the chapter, this is the meristematic tissue. Let's just read or let's just go towards what meristematic tissues. Okay, sorry, let's just go towards the permanent tissue. Okay, what I'm saying, this happens when you are not recording since really long, you are taking off, then this happens. Okay, so this is what permanent tissue. Okay, now we know that permanent tissues will have a characteristic cells. Okay, because they are doing particular functions. Okay, their modifications will also be there in our topic of discussion in this chapter. Now the permanent tissues are what? They could be of two different types. There are simple permanent tissues or complex permanent tissues. What is the difference between simple and a complex permanent tissue? A simple permanent tissue would be the one which is made up of only one type of cells. Okay, it doesn't mean that the cells do not have like in the tissues. When we are talking about permanent tissues or tissues, there will be different tissues coming up, because if you would have understood this concept that from and trying to correlate with the animals what you have studied till now, you said that okay, if there is the cells group of cells present, we call it tissue, means group of cells should be similar, right? Then we said that different group of cells comes together forming the organs. So why the sir is saying that, this point is important to understand. Here we don't say that. We defined that boundary as a simple and a complex tissue. So we are saying that there will be certain tissues which are made up of only one type of cells. But there are complex tissues which are made up of different types of cells. But then why we are still putting it as a tissue? Because if you remember the definition of tissues, what we have said, we said that tissues are those group of cells which have common origin and performing a similar kind of function. So in this complex tissues, they are collectively working together and doing the one common function. So that is the thing. Okay, so this is what the understanding should be. So permanent tissue could be of two types. Okay, what are they? We have simple permanent tissue and we have complex permanent tissue. Okay, simple, complex permanent tissue. Now simple permanent tissue, it is made up of only one type of cells. Okay, made up of only one type of cell, made up of more than one type of cell. So next thing that we have to remember, that what is there in the, what are the types of simple tissue we have to study and what are the types of the complex tissue we have to study. So in the simple tissue we have to study three. What are three? We have parenchyma, we have collenchyma, and we have sclerenchyma. These tissues. Okay, and the complex is what? Complex is two types, broadly classified as what? Vascular tissue. Okay, complex tissue, or it could be also called as vascular tissue or vascular. Okay, vascular is the name given to it, vascular. Okay, not vascular, vascular. Later on people will come and ask that your pronunciations are also not correct. That is what? Xylem and phloem. Spelling mistake, phloem. Okay, next thing is that the xylem. Now the question is that okay, parenchyma must be made up of all parenchymatous cells, collenchyma will be made of collenchymatous cells, sclerenchyma tissue will be made up of sclerenchymatous tissues, but xylem, phloem are made up of more than one type of cells. So what are those? Each of them are made up of 4 different types of tissues. Okay, so let's gonna be remembering that name. So the xylem, 4 tissues that are there, they are what? They are tracheids, there is vessels, there is xylem parenchyma and xylem fibers. Okay, then phloem. Okay, in the phloem we have what? We have sieve tubes, we have companion cells, we have phloem parenchyma, and we have phloem fibers. Okay, easy to remember about the parenchyma words and fibers, which is present in both of them. Tough is tracheids and vessels, steep tubes and the companion cells. Read again and again, you will remember it. Okay, let's just start with the simple tissue. So number one tissue that we will be talking is the parenchyma. Now in parenchyma tissue, this is the most abundant tissue and it has modifications present in the plant body. Means at certain locations they are doing certain functions, so they have modified themselves, different organelles came up and their functions accordingly. Okay, so parenchyma is what? Parenchyma is the most abundant. Okay, so it is the, so we're going to be writing the pointers which we will remember it. First pointer is what? Parenchyma is the most abundant tissue in plant body. Okay, next point that we will remember, that is this parenchyma. Okay, their cells are isodiametric, means they are almost same in diameter. Okay, so isodiametric means cells are what? Isodiametric. Okay, means their diameter is almost same. Okay, so how they will look like? Let's just make it. So it will be like this. This is a parenchymus cells. I just made one type of it. Okay, it has a nucleus present. Okay, we're gonna be making nucleus big, although it's looking too big nucleus, but okay, let's just make it. Okay, now if you see over here, these cells, parenchyma cells, they are not thick walled. Their cell wall is made up of cellulose, hemicellulose. These components are only there, not much is present. Okay, so cell wall is what? It is thin, made up of cellulose, The next thing is, if you see, there is what? There is a space present. Can you see that? This space, so they are what? This is called as intercellular space. So in these cells, there might be intercellular present or absent, depending upon their location. Like at certain locations, like for the hypodermis of certain plants, they are tightly bounded. Okay, at certain places, they are tightly bounded cells. Okay, so there is very less cell space present or no cell space present. Okay, so we will say that intercellular space present or less present. Okay, yeah, so it will be very much present or very less present. Okay, moving on to the shape of the cells. Shape of cell. Now for that you have to remember that SOPE. Okay, what is SOPE? I call it soap. Okay, so as SOAP, soap, is what? This S is for spherical, which I just made. O is for oval, P is for the polygonal, E is for the elongated. So these would be the cells present to it. Along with that, R or the soper. Soper would be a better word. Soper. R is what? Round. Okay, so this is the shape of the cells. Next is what? Where it is present and how different it would be present. Now there are certain modifications of it. So modifications of this tissue takes place. For example, modification. For example, it help in photosynthesis. Photosynthesis. How it help in photosynthesis? In the leaves. They forms what? In the leaf. In leaf, they forms what? They forms mesophyll cell. Mesophyll cells. Now in the plant body, the mesophyll cells could be elongated like palisade type of cells and the spongy round oval cells. Okay, so these are the ones. After photosynthesis, the next thing is there might be acting as a secretory body. Okay, so they can do the secretions. So what types of secretions? Example, tannins, resins, etc. Tannin, resin, etc. So it can do that work also. Then they are having what? Help in buoyancy. So in buoyancy means what? That it help in levitating the plant body. Buoyancy is a feature with respect to what? With respect to the water plants which are submerged under the water. So they are keep on floating because they have what? Air cavities present. Presence of air cavities. What kind of parenchyma it is called as? Aerenchyma it is called as. Aerenchyma. Okay, more to this, what else? It also help in the storage. Okay, storage of the food. Okay, so food, for example, endosperm. Endosperm is what? It is a type of tissue present inside the seed. If you remember the morphology of plant topic, we have done this, that the seed has endosperm which is triploid. Okay, that is also what? Parenchyma cell modifications. These are the basic factors you have to remember about the parenchyma. Let's hop into the next one, that is collenchyma. Now, the first thing, as we have already studied that these features we have to remember. Now, collenchyma is not abundant, but it is present at certain locations. Okay, it's present at certain locations. What would be the locations where it would be present? So, collenchyma is what? The hypodermis of the dicot is made up of this. Plus, it's provide the mechanical strength. So, you have to remember that it provides mechanical strength. Now if a plant is getting mechanical strength, so probably being what? These are those fragile parts which will get affected if they are not present, right? You know that plants can't run for their life, right? But so they need what? They need certain kind of protections. So the outer layer is made up of a layer, like a normal layer, okay, and inside it a layer is present. That is what? That is collenchymatous, because collenchyma is provide the mechanical strength. Now the question is, if something is providing the mechanical strength, then there might be certain properties to it. So these cells would have those properties. We will see that. So they provides mechanical strength to the plant body, slash part, whatever the part it is present. That's why where it will be present, it will be present in the young stem, because in young stem is what? It is very fragile. If you have seen that when the plant has grown, the young stem is young, it will be green also, but it should able to withdraw the effects of the air, winds and all those things, right? So it is present in young stem and petiole of the leaf. Petiole is what? If this is the leaf part, imagine that this is the leaf. It has a small branch which you used to say, that is called as what? Petiole. Okay, we have even studied in the morphology chapter, right? So that if you know that when the wind comes, the leaf flatter like this, but it never get broken off, right? Why not broken off? Because of the strong petiole, and it is strong means it's providing a mechanical strength. How it is providing? With the help of collenchymatous tissue. Okay, moving on. So it is what? Present in the petiole of leaf. Okay, after this the next thing would be what? that how the cells look like, right? So cells we're gonna see. So if I see the cells are what, they would be having certain shape, there had been certain type of cell wall present. Now the cells over here has a large vacuole present, presence of vacuole is there. Okay, next to this thing is what, that their protoplasm is comparatively lower. Okay, because vacuole has taken the most part. Okay, so most of the space. Okay, moving on to the next, that their corners, okay, they will be what, they will be highly thickened. Okay, so if I talked about the cell wall is what, made up of cellulose, hemicellulose and pectin. And at the okay, one point I forget to write over here with respect to this, that it is what present in the dicotyledonous plants. Okay, it's a part of a dicotyledonous hypodermis. Okay, hypodermis of dicot. Okay, it is present over there. We will learn it again, but this is very important fact. Okay, so now these pectin, hemicellulose, cellulose, these are present, and at their corners there are extra depositions of these present, which will give it what, more strength, and that's how the mechanical strength is increased. Understood the assertion reason based question, right? So corners have these material thickenings. Okay, and because of that even the intercellular spaces are obliterated. Okay, intercellular spaces are obliterated. Why it is obliterated? Because thickenings are present. So if I will see them, how they will look like, this is the cell. Okay, now I told you that maximum portion would be what, a vacuole. So imagine that this is the vacuolar part. So this is the vacuole. Okay, and meanwhile this area, this is all covered up with the thickenings. All side there will be thickenings present. Whatever the corners is there na, there will be thickenings present. So these are what, these are the thickenings. Okay, or we call it thickened corners of the cells. Everywhere it will be there. Here also it will be there. Of course I didn't make it, but it will also be there. So like this it will be present. Okay, now come the shape of the cell. So shape of cell. So what could be the possible shape of it? So for it we have to remember about SOP. Okay, so SOP. So we have stopped the E and R. So S is for, we have remembered it for the spherical, O is for the oval, and P is for the polygonal. Clear with this? After this we gonna seeing about that these cells sometimes even has chloroplast. Okay, so in the young stem it is present there. Okay, so some might have chloroplast. So you might have seen that the petiole is what, having chloroplast present, green in color. Young stem is green in color because of this only. Okay, so this thing we have to remember. Okay, the cells will make the food, assimilate the food where the chloroplast would be present. Okay, now intercellular spaces are obliterated or absent means same. Okay, next part is that this is all about the collenchyma. After this comes our sclerenchyma. Okay, so third type of simple tissue is sclerenchyma. when we are studying this sclerenchyma, na, it should come up that this is dead tissue. Why I'm calling it a dead tissue? Because of the thickenings present around itself. So these thickenings are made up of lignin. And when these thickenings are present, lignified thickenings are present, it do what? It obliterates or it obstructs the flow of food and material. So these things becomes what? Dead. Their protoplasm destroy, everything destroy. They are devoid when when they become mature. Because initially any cell that will form, it won't having the thick cell wall. After slowly it will have thickenings present of the depositions which will be secreted by the cell itself secrete it. Okay, nowhere from out it is coming, it itself is making this thing. Okay, so let's start with the sclerenchyma. So in sclerenchyma we will remember what? That where it would be present, that's the most important point. Okay, but that is the types, that what are the types of it, then we will do it. Now the sclerenchyma is considered as a long, thin, cylindrical, thickened cell wall structure. Okay, so they are considered as what? Long, narrow, with thick and lignified cells. Means what? Whatever be the cell of it, it will have a thickening present around itself. See this, how big thickening is present. So this is what? This is considered as lignified thickenings. Lignified thickenings, and which will do what? Makes it is death due to devoid of any material, food and water to this cell. Clear? Now usually they are what? Dead. Okay, so this we will write it, usually dead and devoid of protoplasm. Okay, because later on when they mature, the protoplasm is not present. Okay, now there could be two types sclerenchyma on the basis of how they are look like. They are of two types. Okay, so number one type is that they will be present as a fiber. Okay, so as a fiber, ajo fiber and sclerid.
खाना खा लो चल के।
बनाया तो है मैंने अभी तक।
वही तो कह रहा हूँ, नहा लेते हैं दोनों।
हाँ, बस नहा लेता हूँ। Fiber and sclerid. When it comes to fiber, you know that it will be long, narrow structures. So it will have a tapered ends. So tapered end means what? That both will be fused. This kind of situation. Like the tapered ends are there, so they are going closer. So that's why this is also. So they have what? Tapered ends. Okay, and since they will be lignified also, so lignifications will also be present. But along with lignification, there will be certain pits or spaces present in this. So means they are not completely lignified. These spaces will allow certain molecules to move from here. So these are known as what? Pits. Pits means pore. Through this, substance can travel. Through this, substance can travel. So fibers are like this. And sclerids are what? Circular in appearance and cylindrical in appearance. So they have what? Small lumen like this, and around it a bigger of this would be present. Okay, and they will be what? They will be open to also, because there also there will be what? Certain openings present. So it will be like this. So these are what? These are the pits. This is what? Thickening. This whole area is the lignified thickenings is present. Okay, and this area is what? Lumen. This is also what? Lumen. So you can see that how small the lumen gets over the period of a time, right? So this is what it looks like. Okay, two things. So fibers are what? Fibers are thick walled, elongated, stuck, pointed Okay, so fibers are what? Thick walled, elongated, pointed, okay, and occurring in in group. Whereas sclerots are what? They are spherical, oval, or cylindrical in appearance. Then they are highly thickened dead cells. Okay, with a cavity is called as lumen present. We have seen in the diagram. Now these are commonly, like this is what example given. These are commonly found in the fruit of the plants. Like if you have seen the guava has a grittiness. Why? Because of these sclerots present in its seeds. Okay, so what is what that? So fruit of nuts, guava, like pulp of the guava, pear, and sapota fruit, seed of legumes, leaves of tea. Okay, now what is the major function of sclerenchyma? Sclerenchyma also provide what? Mechanical strength. Because they are dead, they are lignified. Lignification will do what? It will stay there for a longer period of a time. So major thing is what sclerenchyma provides function, if I have to write. So sclerenchyma provides mechanical strength, and it's present in hypodermis of monocot. Okay, where? In the stem part of it. Clear? This we will learn later on again. With this, the simple tissues are over. The next are the complex tissue. Okay, so the complex tissue is also called as what? The complex tissue is also called as your vascular tissues. Okay, now they are vascular tissues means it is broadly classifying two different types of tissues, xylem and phloem. Till now what we know about xylem and phloem? We know that xylem will help in water and mineral conductions from the roots towards the stem and the leaves part, whereas phloem will conduct the movement of food from leaves to the different parts of the body, of the plant body. So it comprises of vascular bundles, that is xylem and phloem. That is xylem and phloem. So we know that what we know? We know that xylem will conduct water and minerals. Conduct water plus mineral. And this conduction is unidirectional. Means from one side, means roots towards the leaves or stems. Okay, so unidirectional conduction, that is from roots to leaf or stem in between. Whereas phloem is what? It conducts food. Okay, and it is bidirectional. So from leaf it is here, it can conduct this to this side also, where the leaf is this side, or it can go toward the roots also. So bidirectional, both the direction it can conduct. So bidirectional or multidirectional also can be said. That is from source, that is leaf, to sink. That could be branch, root, etc. Any part. Flower also. Okay, so anything it could be there. So this is the two parts. Now we have already done that. The xylem and phloem is what they have further parts. They are complex tissues because they are made up of multiple types of tissues coming together doing a common functions. Okay, that's why they are called as what? Complex tissues. Okay, and now let's just start one by one. The first one that we're going to be starting is the xylem. Okay, now in xylems we have four difference of them. Okay, so four parts. What are they? We have tracheids, vessels, xylem parenchyma, and xylem fibers. Now remember that in this, the xylem fibers, they are the ones which are what? They are dead because they are highly thickened ones. So they are what? They are dead. All these three tissues are alive. Okay, so we will remember this part also. Okay, now, oh, wait a second. These are dead. Sorry. Wait. This dead, this also dead, and this is only alive cells. Okay, we will learn about it. Let's go one by one. Now these angiosperms means flowering plants we are talking about. All of these fours are present, but not from the initial points. So when the xylem is present, xylem is what? Certain xylem which will form first and then certain will form later on. So xylem is also two types. If you remember the permanent tissue, primary xylem or secondary xylem. Now this primary and secondary xylem has certain differences present. The primary xylems do not contain fibers. Secondary xylems has the fiber present into it. Further, primary xylem is of two types. One is what? Protoxylem, another is the metaxylem. Okay, so before studying these things, this is the whole thing we have to study up. Okay, so let's just start with the four parts one by one and see them. Okay, so number one is what you have to remember, that gymnosperms lacks vessels, except three of them, like Wilwichitia, Aphytra and genitals. These three are the different orders which contains vessels also. Means they are more evolutionary closer to the angiosperms. Okay, this thing we have already done it in the plant kingdom if you remember. Now let's coming back. So the gymnosperms lack vessels. So means vessels are the characteristic features of angiosperm. Okay, so this will be the characteristic features of the angiosperm. So it becomes what? Characteristic feature. We have already talked about this point. Okay, so this is what? Characteristic feature of angiosperm. After this, one by one, let's talk about all of them. Number one, we talked about the tracheids. So now what are tracheids? They are long cylindrical elongated structures. Okay, they can be tapered at the end or the open could be at the end. So tracheids are what? First of all, we will remember the facts. So first thing, tracheids, they are elongated tube-like cells with protoplasm. Okay, so sorry, without the protoplasm. With thickened lignified walls and tapered ends. It looks something like what? Let's just make it. So let's make it with the blue so that you remember it. They are for the water conduction. Okay, so long like this. So if you see this, this is what long elongated tube like cell, and they have what thickening present. So that's why I made this thickening over here. Okay, and tapered ends. So this is what tapered ends. So everything has been shown in the diagram. Okay, now they have what, they have certain slits present so that things can pass through it. Okay, so they are normally what, they are the dead cells. Okay, they are what, dead cells without protoplasm, because any cell which will be dead means its living constituent protoplasm, cytoplasm, nucleoplasm won't be present. Okay, then the inner cell wall is made up of the inner cell walls have a thickening with a varying forms, and it makes structures like, for example, it is angulated. Like annulated means rings kind of things are present inside. They can be what, it appears like what, septa. So these inside could be septate or aseptate. Okay, moving on to the next thing. So these are the, in the flowering plants, these and the vessels are the main conducting thing, okay, present inside the your plant body, okay, angiospermic body. Then comes the vessels. Now in the vessels, they are what, they are the characteristic feature of angiosperm. Okay, we know about it. Next thing is that vessels are what, individual cells which are stacked together and forms the kind of a vessel's tube kind of a thing. Okay, so individually they are made up of what, individually they are made up of members, individually made up of vessel members. Okay, next thing is what, they are long cylindrical structure, tube like structure. They also have what, thickenings present. Okay, and have lignified thickenings, and they have perforations, obliquely perforations, which is called as perforating plate also. Okay, so those perforating plates or the pits, they help what, they help in the lateral conduction also and simultaneously conductions also. Means individually they are made up of what, imagine that this is one xylem member. So this is what, sorry, not xylem member, the vessel member. Okay, so the next will also stack over here, then next also will stack like this. So individually members stacks together. Now if you see them over here, there is what, there is like this kind of wall present. Of course, every side, that if it is a cylinder will have upper and lower part, right? So how you will remember? You will remember it like this. Imagine that this is the one such vessel. So it is what, it is a cylinder. Can you see that? Can you appreciate this is a cylinder? Now the cylinder will have what, two parts. One is this opening, one is this opening, right? This is the surface area of cylinder. Now if this is the one, so one is here, next will present like this. Now they had to conduct. If this is completely closed, how they will conduct? It won't be able to conduct. So what happened, there is what, this plate which is present over here, it has small holes present, called as pits or the pores. So through that pits and pore, water will conduct. Moreover, pits and pore will be present on this side also, so that it can conduct water on the lateral side also. Means just think now, like inside it a pipe is going in the body of the plant. Imagine that I am the body of a plant and this is going inside from my body till the topmost part. But what about my hands? How my hands will get the water? So pits would be there, through that small paths will be coming out. Parenchyma would be the coming on, that will help in the radial water demands to be completed. Clear with this thing? So these are what? These are perforated plates present, obliquely present, so that what? Water can be passed through it. So water movement would be. Okay. So the next thing is that they are also what? Dead cells. Okay. So that's why they are what? Dead cells. Okay. Next to this is what? And devoid of protoplasm. If they are dead, there won't be any protoplasm present. So devoid of protoplast. After this, the next thing would be that perforations we have talked about. These are what? Perforations through which the water is conducted. Okay. Lateral perforation and pits present for lateral conduction. And who help in this lateral conductions? Other parts of the xylem elements, that are what? That is your xylem parenchyma. Moving on to the next thing. And one thing, that these vessels is what? Characteristic to flowering plants or angiosperm, whatever you want to call it. After this vessels, we have xylem fibers. Xylem fibers also what? They are dead. Okay. Xylem fibers would be what? They are highly thickened cell wall. If you remember the fact little bit, you can write these things. Highly thickened cell wall. Okay. There could be what? Cell wall made of cellulose. Okay. Store foods. No, sorry. They are what? They may be septate or aseptate. That thing, not this. Maybe septate or aseptate, and they are dead because of high thickenings. Okay. Protoplasm will also be not present. Protoplasm absent. This is very easy to remember the facts. Then comes the last one, xylem parenchyma. In xylem parenchyma, what happened? These xylem parenchyma are live, cellulosic cell wall present, thick only, but cellulosic cell wall present. It stored food because xylem also need food. These cells which are closer to present, so it needs food, so they store foods also for themselves and for the other, in form of what? Starch, because that is what plant body, in form of starch only it will be present inside the body. And it helps in radial conductions. We have already said helps in radial conductions. Radial means this side, radius wise conductions would be with the help of this. So help in the radial conduction. That is xylem parenchyma. After this, we have to remember that on the growth phase, primary xylem, secondary xylem would be present. Okay. And what their locations between it. Now there is two types of, like, when the primary xylem is formed, two xylem forms. Certain xylem elements which we have studied, these four forms first. What are they? Xylem vessels, tracheids, and your parenchyma. They forms in the earlier xylems called as protoxylems. Later on, metaxylem is found, which has fibers also. Fibers works are simple, that they provide strength to the plants. Okay, most of the fibers. So primary xylem is of two types, protoxylem and metaxylem. That's we have studied. So classification of xylem on what basis? On the basis, like earlier it was on the types, elements which are present. Now it is on the basis of the development. Okay, on basis of development. First we are doing this. So on the basis of development, we know that the xylem could be what? Primary xylem, or it could be secondary xylem. Secondary xylem due to the secondary growth. Okay, due to secondary growth. Through cambium. Okay, primary xylem would be what? Primary xylem would be of two, like it from a radical, and okay, one thing we won't be writing, from forms primary body. That would be better one. Forms primary body of plant, and it has two types. What are the two types? Number one, we have protoxylem, and then we have metaxylem. So the protoxylem, that is what it contains of three things. It has tracheids, vessels, and xylem parenchyma. Whereas metaxylem will have these three, of course, tracheids, vessels, xylem fibers. With it, there is a presence of, this is xylem parenchyma. These three are present. Along with that, the fourth part, fibers, are also present. Now here comes an interesting twist we have to remember. This proto and metaxylem, how they will be arranged. Okay, so arrangement of proto and metaxylem. Now what happened? This is a difference over here. So what difference is there? That there are two types of it. One is what? Exarch, and one is endarch. What is exarch and endarch? This is with respect to where the protoxylem is present. If the protoxylem is present on the outer layer, means if you look into it and you see that, okay, protoxylem is seen on the outside, metaxylem is seen on the inside, then it is called as what? Exarch kind of conditions. So what is exarch? That the protoxylem is at exterior, that is periphery, and metaxylem at center. So means if you look at certain thing, means for example, if this is the whole body, if on the outer area where my finger you are seeing, if here you see the protoxylem and inside it you see the metaxylem, then we call it what? It is a exarch type of development. Endarch means here the protoxylem is inside or in the center. Okay, so here the protoxylem at center and metaxylem at periphery. Now where we see this exarch is seen in roots, okay, of the plants, where endarch seen in stem of the plants. How we will remember it? Now there is a trick. This is Rx, like your, when you become doctors, then doctors used to write Rx, like at their prescription, write like this, means reaction, right? So what we're going to be doing it? This is our one example. R for what? Roots. X means what? Exarch. It sounds like that, na? What about endarch? This is SN reaction. You might be studying SN reaction in your organic chemistry. So S for stem and N for endarch. not the nucleophilic substitution, right? So this is how we will remember it. This is what about the xylem, everything which we have studied. Now here you have to remember that the primary xylem, initial xylem that is being formed, it is formed by procambion. Later on part is formed by cambium. But secondary xylem is formed by the intrafascicular cambium and fascicular cambium. Okay, so these things we will remember, facts. Okay, moving on to the next thing, that is phloem now. So we have phloem. Easy, same thing the way we have studied, we know about everything. Now with respect to phloem, we have to remember that 4 parts are there, 4 elements are there. So 4 elements of phloem are as follows. So we have what? Seed tubes, we have companion cells, we have phloem parenchyma, and we have phloem fibers. Now earlier what we used to study, we studied that these 3 are dead and this only alive. Now here what happens, that this is alive, this is also alive, only this is dead. So this thing you have to remember, that which will be dead or which will be alive. Fibers are dead in both. Here all the other things are alive, they are only parenchyma alive. Okay, so now, and the reasons also I have told you. Now the phloem transports food from the leaves towards the different parts of the plant body, and they have these 4 structures present. Now always remember that the gymnosperms, they do not have seed tubes and companion cells. In comparisons to them, they have sieve cells in comparison to the seed tubes, and albuminous cells which has a companion cells kind of function. So what we will have to remember? We have to remember that gymnosperms do not have sieve tubes and companion cells. Instead, sieve cells and albuminous cells are present. Albuminous cells are present to perform similar functions. Okay. Now, so sieve tubes and companion cells also becomes what? Characteristics to them. Okay. Next thing is that how the sieve tubes look like. Individually, we're going to be studying all of them. So first is what? Sieve tubes. So basically, they are what? They are the cells, okay, which are devoid of nucleus. So nucleus are not present. So means they are a zombie kind of a cell. Their protoplasm is also very small. Most of the space is occupied by the vacuole. Okay, that is creating a space through that the food can transfer from one part to another. So very less protoplasm will be present, just at the periphery as a thin layer it would be present. Then the question is that, okay, protoplasm present, nucleus not present, they are zombies. So zombie must be controlled by a certain virus. The virus over here, which I will call it what? I will call it the companion cells. Companion cells will do what? It will show the other cells that pathway to do it. So the nucleus of companion cells will help the protoplasm of the, will help the sieve tubes to keep on doing its work. That's why the protoplasm or the nuclear content or the size of the nucleus in the companion cells are little bigger, because it has to do more work. Okay, so this is the one fact that we're going to be studying and we're going to be writing. So sieve tubes are what? They are a long tube-like structure arranged longitudinally associated with companion cells, means companion cells will be present close to it. Companion cells devoid of nucleus, thin peripheral protoplasm present, functions are done or directed by companion cell. Next, large vacuole centrally occupying whole space present. Yeah, and they help in the conductions of the substances. Okay, so that is the mature ones. Okay, so it's everything we have done about the sieve tubes. Then comes the companion cells. Next is the companion cells. So companion cells are what? Their nucleus would be little bit enlarged because it has to do the function of the sieve tubes also. Okay, so they are what? They are specialized parenchymatous cell. Very important. And they are closely associated with sieve tubes elements. Okay, and the sieve tubes elements are connected by the pits. Okay, so associated with sieve tube elements, connected by pits, so that their functions can be guarded. Okay, next thing is that they help in maintaining pressure gradient of sieve cells. Means basically that you know that sieve tubes will be having what food will be coming up. As more food keep on coming in, pressure will keep on increasing. So they help in that passing. When the pressure is more, they pass it to the next cell, the food to the next cell. So their function is like that. Moving on to the next thing, that is phloem parenchyma. Their function is what? They are elongated, tapering cylindrical cells, dense cytoplasm and nucleus. Cell wall is made up of cellulosic cell wall, has pits present, and through that they have what? Plasmodesmata connections. What are plasmodesmata connections? Just for now remember it. The Golgi bodies of the cell, we will study about in the Golgi bodies, they form what? They form a bridge kind of thing between two adjacent cells. That is what? Plasmodesmata connections. So has plasmodesmatal connections with the adjacent cells, and they are what? They store the food as usual, like the plant, so like the xylem. So they store food, okay, as well as help for the radial conduction of the food as well. Okay, food, mucilage and tannin and resins like substances, okay, secretes that also. Next come what? Phloem fibers. What phloem fibers do? They are dead. Why dead? Because they are what? Highly lignified, made up of sclerenchymatous cell, and you know that sclerenchyma is what? Dead. Sclerenchymatous cell means highly thickened cell wall, lignified, devoid of protoplasm. All these things which we have already studied, we can put it over here if we remember just one fact. Moving on to the next things, this is also what primary and secondary phloem. Here also what, in the protophlloem there won't be presence of what, there won't be presence of like in the fibers, okay, phloem fibers won't be present, okay. Same thing is over here also in the phloem, okay. So I'm just gonna be writing it up, summing up is exactly same. So classification of phloem on the basis of development, now they are what, they are of two types: primary phloem, or there would be secondary phloem. You know that secondary phloem will be formed later on, okay, for secondary growth, for primary growth. Now they would be further of two types. What are the two types? Protophlloem, and there could be what, metaphloem. Now the protophlloem will have what, sieve tubes, companion cells, and phloem parenchyma. Here what would be here? Sieve tubes, companion cells, phloem parenchyma, and phloem fiber. So this is seen in case of metaxylem. Their arrangement also would be consist of this thing. Now you have to remember that in this primary phloem consists of narrow sieve tubes. So here sieve tubes are narrow, whereas here sieve tubes are broader, because later on more food has to come up as the plant is growing, okay. So these things are what, the difference between the xylem phloem and the facts which is given in your NCERT. We only did what is given in NCERT in the more concise way in this chapter. If you like, in this topic. So if you like this video, give it a like, share it with your friends, those who are preparing for 2027, those who are preparing for their NEET examination, and those who are just doing their BSc, because these notes can help them in getting up their basic scores clear or the concepts clear. So that's all for this video. Don't forget to subscribe if you haven't subscribed me. I come up with the videos for the NEET examinations which you can take it care for the academics also. Every Monday, Tuesday, Wednesday I am uploading a video for 11th class, whereas for the 12th class I'm uploading on the Thursday, Friday, Saturday. Sometimes on Sundays I'm uploading the shorts or the videos from the 11th or 12th class. Well, that's all for this video. Thank you.